All right, welcome everyone to today's installment of the CNV Web Series. We'll just get started as usual with a few housekeeping things. So you will see on the screen today's attendance URL and QR code. To use the QR code, you just need to open up the camera on your phone and focus it on this, and it should take you to the attendance survey. If you would rather type in the URL is at the bottom, and I will put that into the chat later on in the call in case you've missed it. Next slide, please. And just a reminder about questions, please type your questions into the Q&A box and not the chat box. This is because Zoom keeps a record of all the questions we get through the Q&A box. I will be monitoring the box while Jeff is speaking, and if I can answer the question, I will, and if not, we will mark it to ask him verbally at the end of the call. If we do not get to your question today, we will put it into consideration for our Q&A webinar, which is on March the 12th. And as a reminder, if you have any questions that are not related to today's topic, DGV, please feel free to submit them for the consideration of the March 12th webinar. In your weekly reminder email that usually goes out on Tuesdays, there is a link to a little survey which just asks for any questions that you may have. So if you have any for that, please be sure to go there and send them in to us. So with that, I'd like to introduce Jeff MacDonald. Jeff is a bioinformatician in Steve Scher's lab at the Center for Applied Genomics, located at the Hospital for Sick Children in Toronto. And Jeff is going to be providing an overview of the Database of Genomic Variants, or DGV. So take it away, Jeff.
Great, thanks. So I just wanted to start off by saying thanks to both Aaron and to the ACMG ClinGen CNV Technical Standards Committee for inviting me to talk to you today about DGV. So I'm just going to start off with a general introduction to the database and hopefully provide some insight into the type of data that you can find here, and then follow this up with a review of the DGV Gold Standard resource. So the database was initially launched back in 2005 following the initial description of copy number variation in unaffected individuals. And as more data became available, the database was expanded and updated to accommodate the increased number of studies that were publishing CNV data. So the objective really was to provide a comprehensive summary of structural variation in human genome, defining this as genomic alterations of DNA that are larger than 50 bases. And important to note that this database contains structural variation identified only in control healthy individuals. And we really wanted this to provide a useful catalog of control data for studies that were aiming to correlate genomic variation with phenotypic data. And it's continually updated with new data from peer-reviewed research studies. So again, specifically, the aims were to provide this information about variants identified in control samples, serving as a resource for both clinical and research labs. And we wanted to be able to show this variation in a genomic context using a genome browser to make it available at no cost and to provide access to all the underlying source data. So the database has been recently updated, as I mentioned, and contains information from about 75 different peer-reviewed studies. So currently there is approximately 980,000 different CNV regions and just over 4,000 inversions that have been identified in just over 47,000 samples. The majority of the data that's in DGV are from 4 different types of studies. We have data primarily from array-based CGH and SNP arrays. So microarray-based studies. We have whole genome sequencing using both next-generation sequencing technologies and Sanger-based. Data from clone-end sequence mapping and sequence trace mapping, which is also derived from Sanger-based approaches. And to consider as well, so the database is still limited in content. The amount of data that's becoming available is still growing. There's only approximately 47,000 individuals that are represented right now, and these are not ethnically matched. And due to biases in some of these studies to date, the content is almost certainly not an accurate representation of the structural variation on a population scale. And these biases are somewhat historical in nature and are related to the sample selection and inclusion. So there's definitely an overrepresentation of Caucasian or European individuals. And as a result, the database may not have an accurate representation of all populations. And I will touch on this again when discussing the DGV Gold Standard, and I'll actually show a population breakdown. So as mentioned, the database was recently updated and is now available on our site. We will work with UCSC to get this updated, available as soon as possible. And with this update, we have made a version of the DGV Gold Standard available for the HG38 assembly. And we've also added 3 new studies. These studies include the Nomad SV dataset under accession NSTD 166 at DBVAR. And there's 2 other studies as well, which in total have added an additional 10,000 new samples and just over 400,000 new CNVs. So for anyone who hasn't used the database before, or who primarily accesses the data through either a third-party software or through the UCSC Genome browser, I'll start off here with a very quick introduction to our site and highlight a few resources that are available and the corresponding access points. So the URL for the site is listed at the top of the page, and I've also included a screenshot here of the homepage. So there are links at the top to access the Genome browser here, where you can view the CNVs for your genomic region of interest. And we have links to access the full data sets on our downloads page here. There are some tutorials and answers to frequently asked questions here at the links on the right-hand side. And we have also implemented a table browser where you can selectively search for entries within individual studies, search for variants from specific samples, or you can display all the CNV found in a certain region. And you can access that here through the query tool or through any of the links here. So just a quick view of the Genome browser. So we have data that's been mapped to HG18, HG19, and HG38. And you can select the corresponding assembly from the drop-down menu here. You can use the select track option at the top over here to determine which data sets and annotations you wish to view. And here we see a list of all the different variants in our DGV structural variants track as an example. And I just wanted to show a quick snapshot of the table browser. So you can see that there are multiple tabs here for each variant attribute, listed by study, a list of the variants by sample or different approaches. And you have an option to filter these up here. And if you wish to search by multiple criteria, you can just select the plus option to create additional filters. So I just have a couple screenshots here of the DGV structural variants data, showing a region in the Genome browser where we have a single duplication here in blue and a number of overlapping deletions from several different studies that are highlighted in red. Clicking on any of these entries will pull up a detailed summary page that has additional information about the variant. An example over here showing the variant length, number of gains and losses, the allele state if the variant had been genotyped. And we've also provided links to other assemblies in DGV. And we also have links out to the UCSC and the Ensemble Genome browsers. So I won't go into any more detail here, but as mentioned, there is a tutorial available in the training materials section, the link available from the homepage. And that goes into quite a bit more detail on the functionality and the options that are available in the Genome and the table browsers. So we often get questions about the various CNV data sets that are available and how they differ. So I'm going to spend just a bit of time introducing these and outline what each contains and how they differ from each other. So the DGV structural variants and the supporting variants are the primary data sets that make up the content in DGV. So there are currently 75 peer-reviewed studies, each of which is included separately, is accessioned by the group at DBVAR, is curated by us, and then loaded into DGV. In the supporting variants tracks, each variant here represents a single structural variant that's been called in a single individual in a single study. So these can often be referred to as sample level or supporting level calls. And to note, so there's currently just over 7 million entries here. And as a result, the display can be extremely verbose and it can be difficult to view and interpret. So these are provided only as a resource for those who may be interested in looking at the raw data. But for the most part, this data will not be overly useful for most individuals. So the primary DGV structural variants track contains the structural variant regions, where a representative structural variant model has been created. And this is to represent the overlapping sample level calls for any particular interval. The regions are created per study, and how these regions are generated will depend on the process used in each individual study. So in most cases, the authors would require that a minimum percentage overlap of each supporting variant is required in order to assign them to the same region. Some will use a very strict threshold of 100% overlap, and others may be relaxed as requiring maybe only a 50% overlap. And lastly, the DGV Gold Standard Variant Track contains a curated set of CNV regions where all of the underlying data from a subset of high quality studies are combined together to generate a consensus cross-study set of variants. So now I'll go into a bit more detail on the DGV Gold Standard Dataset. So the motivation for creating this dataset was twofold. The first was to increase the sensitivity by creating a comprehensive reference set of variants by leveraging these large, unique sets of samples that exist across multiple studies. By combining a number of different complementary approaches will help to better define the underlying variant as well. And we can improve the power to detect variants across diverse genomic regions and across different size ranges, compensating for the individual study biases and shortcomings. The second was to increase the specificity. So we really wanted to tackle the problem of having false positives in the data set. So we wanted to be able to remove these using support from multiple samples and multiple approaches. So look, if we look currently in DGV, the variants span over 85% of the genome, which is most likely an overestimate. So we really wanted to improve the resolution and the accuracy of the variant breakpoints. And we also wanted to create more accurate frequency estimates and provide a broader population representation at each variant region. So the approaches that were used for generating this resource are outlined in the paper that's listed at the bottom here. And feel free to take a look here if you want more details on how the dataset was actually put together. So here I just have a brief summary of the workflow that describes the process. So we started really by taking all the current data sets that were in DGV. We removed those older low resolution studies. We then evaluated the remaining entries using the process that was developed in the article I mentioned earlier. So in general, we identified studies which potentially had a high false positive rate by looking at the validation rates in each of the individual studies. We also looked at studies where there were a disproportionately large number of singleton calls that were found only in a single sample, even when a large number of individuals were tested. And we also looked at studies that had a high number of variants identified in regions that should be under constraint. So after going through this process, it left us with 32 different studies with variants that were called in approximately 23,000 unique samples. So at this point, we set out to determine how much support would be required from the combined data to be considered a gold standard variant. So this plot here shows the percentage of the genome that is variable when various combinations of support from samples and studies are combined. So we see that a large proportion of the genome is variant when only one sample and one study are required. And this drops quite quickly when we require support from multiple samples and multiple studies. So the decision was ultimately made that in order to reduce sample-specific artifacts, we required that at least 2 different samples contain the variant to be included. And in order to reduce any study-specific biases, we required that at least 2 different studies called a variant to be included. So in summary, we end up with a data set of just over 38,000 variant regions when all the data is combined across these studies. So in contrast to having over 85% of the genome spanned by CNVs in the entire DGV dataset, the gold standard reference has refined this to an estimate of about 17%. And by combining the data across multiple studies and requiring support from 2 or more samples and 2 or more studies, we've achieved a robust representation of CNV in the genome. In addition, combining data from over 23,000 samples allows for a more accurate assessment of the CNV frequencies and allows for interpretation at the population level. By restricting the input to high resolution and high quality data helps to minimize the errors and false positives. And just a quick note, looking at the table here, I'll just outline that we do see a bias in the number of losses and the number of duplications that are detected. And this is a direct result of the detection bias in the individual studies used. Deletions are in general much more easy to detect than duplications. So I've just provided a table here, and this summarizes the data sets that were used with their corresponding accessions from dbvar, and then just a brief summary of the approach and the methods that were used by each. So as we saw in the flow chart earlier, the original DGV datasets were evaluated for quality to remove older, noisy studies with a high false positive rate. Also, in order to improve the breakpoint accuracy, we selected studies that could be considered high resolution. So we selected sequencing-based approaches, both Sanger and NGS-based. We also selected array platforms that had a minimum of at least 1 million probes. And last, we included a couple studies that used custom arrays that, although they had less than 1 million probes, they had a high probe density at known CNV loci, which would be beneficial when trying to refine the CNV breakpoints. So I just also wanted to briefly go over how the regions were generated and how we assigned the breakpoints. So the DGV Gold Standard Variant regions were created by identifying overlapping CNV calls and combining these together to form a representative region. We used a clustering program to build these calls where each underlying variant shared at least 50% overlap with all other members in the same cluster. So this was done in a bidirectional manner to remove any clustering bias. The resulting region was then created where the maximum number of entries in each cluster was achieved. So what we do see in some complex regions where there is a large number of entries, you may still end up with multiple overlapping calls, which may be inevitable, unfortunately, given the large number of entries with varying breakpoints that are derived from a multitude of different approaches and platforms. So I've included a schematic here of a relatively simple region to highlight the process. So we see a number of overlapping deletions here with differing breakpoints, but all of which are quite similar. Each of these will share a minimum of at least 50% overlap with each other. The resulting gold standard CNV is depicted at the top. And we define the inner boundary of this variant here by selecting the start and end coordinate of the CNV with the highest breakpoint accuracy. So each platform and method combination was evaluated and assigned a score. And as you can imagine, a Sanger-based sequencing approach will capture the breakpoints much better than an array, and would be given a higher rank. So we can see here as an example, the SSV123 has the highest rank, and the inner coordinates of our gold standard entry are defined by the start and end of this variant here. The outer or maximum boundaries are defined by the maximum span of all the underlying variants that were used to generate this model. I won't spend much time here, and the table may be difficult to see, but I just wanted to include a quick example of some of the platform method combinations and the ranking that was used or assigned to each for determining the breakpoint coordinates. So we can see a combination of different sequencing platforms and different algorithms or approaches, and we have a rank that is assigned here. And for each variant in the DGV set, we'll actually display the ranking that was used, so you can use that to help determine how accurate the breakpoints may actually be. So when determining the CNV frequency, we couldn't just use the total number of unique samples used in all the studies, as not all the studies and not all the approaches have the ability to detect variants at all regions across the genome due to variations in coverage or representation for each particular approach. So to calculate the frequency, we identify which studies had the power to detect variants at each region individually, taking into account differences in coverage and resolution for the various approaches that were used, the number of unique samples that were reported to contain a variant as a fraction of the number of unique samples that were actually tested at each locus was used to determine the frequency. And the redundancy in the common cohorts and the samples across the various studies was addressed and taken into consideration. So looking at the gold standard dataset as a whole, and as mentioned in one of the earlier slides, we do see a bias in the number of deletions versus the number of duplications. And I've highlighted the number of CNVs of each type in each size bin. So the variants in the smaller size bins are predominantly derived from sequencing-based approaches. And this also explains why there are significantly more deletions here than gains or duplications. We definitely start to see an increase in the number of duplications in the larger size bins, for which array-based approaches are contributing. So in total, there were just over 29,000 samples that were used in the studies that were selected for inclusion in our gold standard set. A number of these studies use the same cohorts, and therefore there is some redundancy in the samples that were used. The 1,000 genomes, the HapMap, and the Human Genome Diversity Panel samples are the most commonly replicated samples. And after accounting for this redundancy, we end up with just over 23,000 unique samples. If we look at all the unique samples that were included, we see that when the sample identity is known, the vast majority of these samples are of European descent. So there have been concerted efforts to improve representation, and we have seen an increase in samples from other groups, but there are still more samples needed from these and other individuals in order to have a more balanced representation. We see over here in gray as well, in the greenish color here, there's still a large number of samples for which we do not have any information. And these will represent samples where the study authors either did not collect this information or they did not have consent to release this. So I just wanted to show the data in the DGV Genome browser to illustrate what the results look like. We can see a large number of entries in the primary DGV structural variants track here, with a few large duplications across the top and a number of overlapping clusters of deletions and duplications below. At the top, we can see the DGV Gold Standard data track, showing 6 different entries across here. So the large duplications here at the top were excluded, as 2 of the studies didn't pass the initial QC for inclusion. And we also have overlapping calls from the same study here, the Cooper 2011. And as we require support from at least 2 different studies, this large duplication is not represented in the gold standard. We see a cluster of overlapping deletions here on the left, and the corresponding entry in the gold standard above. There are a few gold standard entries where we have many overlapping calls, like in this example here. And there are some where there are only 2 different studies. So if we look at this example here, there's only 2 different studies here that actually call the variant. So this highlights that although all the entries have passed the minimum thresholds, there is still substantial variability in the amount of support that is available for each gold standard call. So by clicking on the entries, a summary page with all of this information is available, allowing you to further evaluate the entry. So I've zoomed in on a particular example where there's support from a number of different studies. The gold standard variant is included up here at the top. And if you mouse over the variant, a pop-up window will appear and provide a brief summary. So here we see the variant ID, the number of sample level variants that were used to generate the reference variant. A total of 149 unique samples were found to contain this deletion. We can quickly note that the difference in the number of samples and the number of variants is a result of these common samples that were used in multiple studies. And we see a frequency of the variant reported here as well. So with a total of 149 samples with the deletion, we'll see on the variant detail page that there was a total of 16,000 unique samples that were tested in these 9 studies, giving us this frequency estimate of 0.92%. And if you would like additional information, you can just click on the gold standard entry and that variant summary page will pop up. So this is a section of the variant summary page. I'm going to highlight a few items that may be useful. So as mentioned at the top here, we have the variant frequency, which has been identified. And we have a breakdown of a population summary here showing what groups the CNV was found in. We also have a list of all the different studies here for which the CNV was called. So we see 9 different studies that were listed here. As we scroll a little bit further down the page, we have information available on the number of samples, the different tools or programs that were used to call the variant. We also have the total number of unique samples that were tested. And that's what's used to calculate the frequency, as mentioned earlier. And we've also displayed the inner rank here, which will give you some insight into the quality of the breakpoint assignments. So as mentioned, a score of 1 is the highest and a score of 16 is the lowest. So this may provide useful if there is some discordance between the boundary of this entry and your own result. So I just want to mention as well, in addition to hosting the DGV and the DGV Gold Standard data at our own database, we have also made this available at the UCSC Genome Browser. So to note as well, while the structural variant and supporting variant datasets are available for both HG19 and HG38 at UCSC, currently only the HG19 version of the gold standard is available here, and we hope to have this updated soon as well. And again, to display any or all of the data at UCSC, you just need to navigate to the track settings page and then click the appropriate subtracks over here at the top of the screen to select which data sets you'd like to look at. And lastly, at UCSC, I've just pulled up the same region that we viewed earlier in the DGV database. I just want to highlight that the format and the content is almost identical, allowing for ease of use across the different websites. So here, I've shown part of the data from the DGV structural variants track for a complex region. And we'll just provide an overview of the gold standard output and some suggestions on how to interpret this. So we can see that some regions may contain a large number of these overlapping CNVs, and it may end up resulting in overlapping gold standard entries as well. And this may result from a couple different things. It could be the underlying genomic complexity. This region may be rich in segmental duplications or repeats. And the overlapping entries in the gold standard may also be technical in nature. We may see that there is an inability to resolve variants based on the differences in the platforms or approaches that were used. So I've scrolled further down and you can see the DGV Gold Standard variants in the middle here. So there's a number of overlapping duplications and deletions. So I'm going to focus just on the deletions for this example here. And in addition, I've loaded the probe files for a number of commonly used array platforms, many of which are used in the studies that were included in the DGV Gold Standard. So as we can see, there are gaps in coverage here in the Affymetrix array platforms, in the GSV L118130, and we also see gaps in the left-hand side over here in some of the Illumina platforms. So as a result, the underlying variant calls here may differ enough that they will not meet the minimum reciprocal overlap threshold and could be split into two distinct clusters. So this variant over here to the right is relatively unique. And this variant over here extends quite a bit over to the left, and it may be unique as well. If we look at some of the summary details for each of these, we see that this one over here to the right is found in only 7 samples, all of African descent and at a very low frequency. So this may in fact just be a rare population-specific CNV. The 2 variants in the middle are quite similar, and they've both been detected in a relatively large number of samples and studies. So we have just about 400 samples in one and over 2,000 samples in another. And the frequency estimates for both are relatively similar. We have about 12 and 11%. So these are therefore most likely the same call. And because there is a lot of evidence, we end up with an accurate frequency estimate, even though they haven't been combined. To quickly note, if I went back and actually looked at these manually and combined the data, the total combined frequency is 12%, which is concordant with both of the individual reports. So the take-home message here is that if there is enough evidence, the frequency estimates should be accurate, even if there are overlapping entries. And finally, the entry that extends quite a bit further to the left here was found in over 500 samples, but has a significantly lower frequency and may actually represent a different biological variant. So after looking at a couple of specific examples, I just wanted to quickly review the data set as a whole to provide an overall summary of the content. So we see that there are a large number of entries that only meet the minimum threshold for the number of studies that support a CNV. This is likely due to a number of factors, the first of which is that there is an inherent difficulty in combining data that was detected using different approaches. And secondly, we do see and expect that a large number of the variants will be relatively rare, And as a result, we wouldn't expect many different studies and approaches to detect the same rare variants, especially using different sample sets. If we then look at the distribution of copy number variation by size and by frequency, we definitely see an overrepresentation of rare variants. And in general, we see a larger number of small variants as well. The enrichment of these small variants is being driven by the NGS sequencing-based studies, which on average will detect a much greater number of variants per sample, and will also have the resolution to detect much smaller CNVs. We do see this anomalous peak in the 99 to 100% window as well, where we see an increase in the number of structural variants in this category. And these are potentially problematic and likely either represent instances where there is a reference artifact and all the samples in the study are reporting a variant, or we may see variants with minimal levels of support in studies which only analyze a small number of samples and have therefore erroneously elevated the calculated frequency. And so to highlight this, I have just plotted the number of CNVs at each frequency bin with the average number of samples per CNV in each. So as expected, we see a small average number of samples in the rare bin, and this continues to rise as we approach the 75 to 90% area here. And then this drops significantly in the 99 to 100% category. And this kind of supports the idea that this category tends to be enriched for variants that are found in these studies that have only tested a small number of samples and for which the variant was found in all those samples tested. So a few things to consider. So the category that appears to be of the most concern are the extremely common calls, those found at a frequency greater than 99%. And there is that possibility that these may be reference artifacts or false calls. When looking at this category in a bit more detail, we want to look to see if we could find any specific patterns that would emerge. And when we do this, we do note that the majority of these are small variants. They're usually less than 10 kb in size. Most of these are also only found in 2 subjects in 2 studies, which are meeting the very minimum inclusion criteria. And lastly, we also note that many of these reside in repeat-rich regions. So where over 75% of the CNV length is actually annotated as complex. So it may be simple repeats, entries in repeat masker, segmental duplications, satellite repeats, assembly gaps, things along those lines. And so just to note then, CNVs in this category should be evaluated with caution. So I also did a little exercise and wanted to check and see how often we have DGV Gold Standard variants that appear in regions where we would not expect to find these CNVs in healthy controls. So to do this, we took the list of dosage-sensitive genes from ClinGen, extracted the deletions which overlap these genes with evidence for haploinsufficiency, and compared the duplications that overlap genes with evidence for a triple sensitivity. We then looked at the general characteristics of the regions and the variants to see if any patterns would exist, and if these patterns could inform us on what criteria and thresholds would be appropriate when using the gold standard resource. So initially we detected 60 genes which overlapped either in whole or part with one of the deletions in the gold standard set. Many of these were found in repeat-rich regions, which would be commonly removed from further analysis. If we remove those variants, which had more than 75% of their length covered by repeats, we end up with just 53 genes. If we then look at the gold standard variants and remove those that had a frequency below 1%, so those rare variants, and if we remove variants that are problematic in that region that were almost 100%, then we only have 7 genes remaining in our list. And then the last option would be to look at the entries and see how many of them have only met the minimum requirements of having 2 samples in 2 studies. And if we remove these, we're left with 4 genes, and I have them indicated here in this table. Lastly, if we require more than 2 studies and or more than 2 samples, we don't find any that overlap CNVs. We had a much lower overlap with the triple sensitive genes. So we see 1 entry here, and this is found at 0.02% frequency, which is quite rare and would likely be removed anyways. I was also asked just to give a brief overview of how the DGV Gold Standard Dataset compares to other population CNV data. So I've used the Nomad SV resource here that Ryan spoke about last week as an example, and I'm by no means an expert on this data set, but we'll try to provide some general insight. So I've used the data that was available at DBVAR under the accession NSTD166 and information from the Nomad SV website for this table here. So there are definitely a number of similarities between the two. They both include data from a large number of samples and across multiple populations. The SV calls have been annotated to include accurate breakpoints and frequency estimates. The majority of the differences lie in the underlying data, including the samples that were used and the approaches that were used to call these variants. So quickly, just to note, so there's about 23,000 samples used in DGV. We have about 11,000 in Nomad SV, and there's actually no overlap in the samples that were used for both of these, so they are unique. We see that there's multiple different microarray platforms, NGS and Sanger-based sequencing, although at low coverage, low to about 15x coverage in the Genome of the Netherlands study. While in Nomad SV, they have high, about 30x coverage on average, using only NGS-based approaches. So we see that we also look at the types of variants that are being described in both. There's various different SV classes that are described in the Nomad SV dataset. And currently in the DGV Gold Standard, we're only representing gains and losses, deletions or duplications, but hope to increase this to be more representative of the data that we actually see in DGV. So looking at the data sets as a whole, looking at the size distribution, in general, we see similar trends where the number of entries declines in the larger size bin, although the DGV Gold Standard does have a proportionally increased representation in this 1 to 5 KB window here, and in the greater than 50 KB size window over here. So the majority of these are most likely detected using microarray-based approaches, which are well suited to detect these larger gains. But we do see a large number of entries in the Nomad SV as well. So in general, I would suggest that both data sets provide valuable information that will be complementary in certain regions and in certain sizes and could be used together. As each data set continues to grow, we'll likely see much more concordance in the content, but in the interim, it would seem to be beneficial to use both. I just wanted to quickly look at the reported frequencies. So looking at an example here that's detected in both the DGV Gold Standard at the top and in Nomad SV, we see that there's a difference in the reported frequency, even though a large number of samples is reporting the variant in both databases. So we see a frequency estimate of about 13.5% in DGV and about 5.7% in Nomad. We have just over 10,000 samples reporting here, and we have just over 3,400 samples reporting here. So at this point, I think it's important to highlight the differences in how the frequencies are calculated and what they actually represent. So the variants in Nomad SV have been genotyped, and therefore they've identified which samples are heterozygous or homozygous, and are therefore reporting the allele frequencies. The vast majority of the data that was used to create the gold standard was not genotyped. So we only have a count of the number of individuals that have a deletion at this region. So therefore, the frequency calculation actually represents a variant frequency and not an allele frequency. And in addition, the variant frequency is actually reported as a percentage here, whereas the Nomad SV frequency is not. So when we actually go to the Nomad SV browser and look at this entry, they actually do report the number of homozygotes. So we can therefore determine the approximate number of individuals that were found to have this deletion, and then calculate a variant frequency that's similar to DGV in order to compare apples to apples. So after performing this conversion, we can see that the variant frequency is about 11%, which is quite similar to the value we see in DGV. And so as it turns out, both data sets actually support the same result. So there may be other situations where the frequency estimates are discordant, and this could be due to a number of factors, either biological in nature or technical. The sample sets and populations represented are different, so this may account for some of the differences. And in addition, there's also differences in the platforms and technologies that are used, and the sensitivity to detect variants may impact that frequency estimate as well. So if anyone is interested in obtaining additional information on the DGV and the DGV Gold Standard Datasets, the following publications highlighted here will provide a bit more in-depth description of each. And here I've included the website address again at the top and also our contact email. Please feel free to reach out if you have any questions about the database, and I'd be more than happy to help. And I will end off here and I'll turn it over to Erin.
All right, thank you. If you will stop sharing, I can start sharing. All right, get this pulled up for you here. Okay, so now we will switch gears and look at an example CNV with population data. This is a duplication at 19p13.11 in a 2-month-old female with tetralogy of Fallot, and inheritance information is unavailable. Now, this duplication is only about 19 kb in size, but just for the sake of example, just please assume that this is something that you could reliably detect by your platform and meet your reporting requirements. So since this is a duplication, we'll begin by using the gain scoring metric. So walking through each section of the metric, the first thing that we do is an initial assessment of genomic content. We can see that this duplication does include at least 2 protein coding genes, so we'll apply category 1A, which is worth 0 points, but continue our evaluation. The duplication does not overlap any established, as in curated by ClinGen dosage sensitivity, dosage sensitive or benign genes or genomic regions. And again, the CNV only includes 2 protein coding genes, so we will apply category 3A, which is worth 0 points, and continue our evaluation. So this brings us to section 4, which is the detailed evaluation of genomic content. When we were looking at the CNV in the UCSC browser earlier, we saw that there was indeed a DGV Gold Standard duplication very similar in size to our CNV. In this case, this might be a good place to start, as opposed to looking into the literature for the 2 genes involved. If this population variant indicates that this CNV is benign early on in the process, it may change the way we approach any subsequent literature evaluation. So let's look into this DGV Gold Standard variant. So as we talked about earlier, you can click on the variant in the UCSC browser, and that will bring you to a details page that looks like this. In the red box, I'm highlighting the overall frequency of this particular variant as reported by DGV, which is 12.9%. And this is, of course, much higher than the 1% threshold we put on category 4.0, the population variant category. So given this information, should we go ahead and apply the -1 points and classify this CNV as benign? Not so fast. So let's take a look at the data that's underlying this 12.9% frequency. So this variant has this frequency estimate because it was observed 4 times in a total of 31 samples tested. This is a pretty small number, and it brings into question how accurate this 12.9% estimate may be. So when evaluating population variants and calculated frequencies, consider guidance put forth by the ClinGen Sequence Variant Interpretation Working Group regarding use of the BA1 sequence variant evaluation criteria. The criteria in which, if met, allows the variant to be classified as benign with no other supporting evidence. In the paper that I'm referencing at the top of the screen, the group recommended that the threshold allele frequency be observed in a general continental population data set of at least 2,000 alleles before this criterion can be applied. This is a good rule of thumb for our scenario as well. So going with this recommendation, the variant we observed in DGV by itself shouldn't be used to call this CNV benign. So let's just check and see if we see any similar events that have been observed in Nomad SV. And when we input our coordinates here, we also observe a duplication very similar in size to ours, marked in the red box. When we click on this particular variant for more detail, we see that it seems like a high quality variant observed in several different populations across many different adult age ranges, and has even been observed in 4 homozygotes. The overall allele frequency for this variant is 0.36%, which is below our general 1% threshold for category 4.0. However, when we look at the population breakdown, we can see that it's at a much higher percentage in the East Asian population, almost 3%. This is coming from a continental data set with at least 2,000 alleles tested, so technically that would be appropriate for consideration of category 4.0 at the full -1 points. You might ask yourself why this would be appropriate if we do not know the ethnicity of the patient, but the idea here is that if a variant is at this high of frequency in any population, it's really unlikely to be causative of Mendelian disease in any other population. The ethnicity of the patient doesn't matter here the way that it might if you were trying to determine that the variant was absent, but you hadn't adequately tested the patient's ethnic group. However, for the sake of example, again, I'm going to show you another way to think about this if the numbers didn't work in your favor like they happen to here. So say you only had that overall frequency information, that 0.36% allele frequency. That is, again, technically below our 1% threshold. But remember, that 1% threshold is a general guide. Just as it is for BA1 in the sequence variant guidelines, that particular number is not going to be appropriate in all circumstances. So for some phenotypes, particularly those that are very rare, this number is going to be too high. And in some situations, it might be too low. For certain diseases and phenotypes, enough information might be available to calculate the maximum allele frequency of a disease-causing variant in the general population. You can use the CardioDB allele frequency calculator, which I'll show you on the next slide, to do this. In general, what you're trying to do is be conservative. If your actual allele frequency exceeds the highest plausible maximum you can come up with, you can feel comfortable awarding the -1 points and calling the variant benign. When using the calculator and trying out different numbers, again, which we'll go through momentarily, try to put in, the highest reported disease prevalence that you can find, the most frequent single gene cause that you can find, and the lowest credible estimate of penetrance that you can find, again, in order to get your most conservative numbers. So here's that calculator. Let me show you how this works. The URL for the calculator is at the top of your screen, and you can read more about it in the 2017 publication by Whiffen et al. In Genetics and Medicine. By entering a few pieces of information into this calculator about your phenotype of interest, you'll get both a maximum credible population allele frequency and a reference allele count that can be tolerated. Now, remember, our phenotype of interest here is tetralogy of Fallot. So assuming that this is inherited in a monoallelic fashion, I put in a prevalence of 1 in 3,000, which was information I obtained from NORD after a quick Google search. The website itself, the calculator website, has helpful guidance on what types of things to put in the allelic heterogeneity and genetic heterogeneity fields. And in the interest of time, I'll let you read more about that there. But if you do not have a firm estimate on the exact number of genes that causes a particular disease, which is the genetic heterogeneity, or the maximum proportion of variation within a gene that's attributable to a single allele, and that's the allelic heterogeneity, you can do what I've done here, which is to set the genetic heterogeneity at 1 and use the allelic heterogeneity to convey the most frequent known cause of this particular phenotype. So in this case, per a 2010 review study, the most frequent cause of tetralogy of Fallot was the 22q11.2 deletion syndrome, which was the cause for about 7% of patients. Another way of thinking about this is that it would be unlikely to discover a cause for tetralogy of Fallot at this point in time that was more frequent than the 22q11.2 deletion syndrome, as we would have likely already discovered it by now. Again, for the sake of example, I'll leave the default penetrance where it is at 0.5 for now, and I changed the reference population size to be the number of samples tested at our locus of interest in Nomad SV. With all of this information that I put in, you can see that our maximum credible allele frequency is 2.3 times 10 to the -5th, and our maximum tolerated reference allele count is 2. To refresh your memory, the Nomad variant we were interested in had an allele frequency of 0.003 and an allele count of 79, which is much higher than these maximum credible estimates that we're getting here. So what if we were unsure about one of our variables? So let's see what happens. Let's see what happens if we change the penetrance here. If I bring it all the way up to 1, our maximum allele frequency and count goes even lower. If I bring it all the way down to 0.01, our maximum credible numbers go higher, but they are still lower than what we were actually observing in Nomad SV. So in this particular case, because of how rare tetralogy of Fallot is. No matter how you fool with these variables, they're still going to be lower than what we're actually observing in NOMAD. And you can use this tool if you have some idea of the prevalence, the most common genetic cause, and the penetrance. And you can use the sliders to see how any one variable affects the N maximum credible allele frequency. If you have a situation like this where any way you manipulate the numbers, your population variant is still too high, you can feel more confident awarding the full -1 point, even if the frequency is not technically at that 1% general generic threshold that we put forth. This will not always be the case, and you won't always have the information that you need in order to be able to use this, but I did want to show it to you for those times when it might help you figure out the best course of action for a particular variant. So to summarize, in this case, it does make sense to award the -1 point, so using category 4O, the population variant category. And though the overall allele frequency was below 1%, the frequency in the East Asian population was almost 3% with over 2,000 alleles tested. This alone would have been compelling information. However, again, because we're just doing an example here, we want to step further and show that given the rarity of this phenotype, even an allele frequency of 0.36% was too high to make sense as a possible cause for this condition. So even though the variant that we saw in the DGV Gold Standard Dataset indicated a frequency of 12%, we didn't want to rely on this information alone because it was made with a small sample size. But note that, as Jeff just told us, DGV does have many other variants in the Gold Standard Dataset with appropriately high sample sizes, so always be sure to check on that information. To complete the evaluation, do a quick literature search to ensure that there are no reports of a phenotype associated with whole gene duplications of MPV17L2, or with the potential loss of RAB3A, which was the gene that was potentially disrupted by the duplication. Assuming none, the CNV can be classified as benign. So at this point, we will take questions, and I'll leave the attendance code up as we go through some of the questions that you have sent in. So give me a moment to find the Q&A box and start asking the questions. So Jeff, I know that you mentioned at the beginning that DGV has just incorporated NOMAD SV, but is that just in the general DGV data? Has it been incorporated into the gold standard set yet?
So the NOMAD SV data is only included in the primary DGV dataset now and has not been included in our gold standard. So we're hoping to have the updated gold standard available relatively soon, but it's not quite there yet.
Okay. And then another question, when you are downloading or browsing the DGV Gold Standard data, is it possible to filter the data to only include calls that were made using a minimum ranking? So maybe only those studies ranked less than 10, in other words, the most highly ranked studies?
So I'm not sure if the minimum ranking here refers to the breakpoint assessment. But if so, currently we do not have the option to filter the data based on a number of those different criteria. But that's definitely something I can see would be useful, something we can work towards incorporating. We do have the data available for download as a whole, and it's not too cumbersome. So if you were interested in just downloading the data, you could play around and just look at the data that meets the criteria that you're looking for as an option in the interim.
Okay, next question. Someone was curious why NGS-based approaches seem to detect deletions more easily, and is this generally true?
So yes, in general, it is true. And it's going to depend on a number of different factors, primarily the different approaches that were used to call the variants. So if you're using like a read depth-based approach, it's going to perform very similar to a microarray where if you have a deletion, you're going to see a 50% drop in your signal intensity, which is much more striking than if you have heterozygous duplication, where you have a marginal increase in your read depth. At the same time, if you're using like a split read-based approach, in order to capture an insertion, it has to be small enough to actually fit within the read that's being mapped. Whereas deletions, you have a split read that spans a much larger regions. So you're going to detect many more deletions in that case as well. So there's a few different technical reasons why in some of these classes and these categories you will find a little bit easier to call deletions, and that's why we do see a larger number of these in the data sets.
Okay. Someone also asked if I could please clarify category 4.0 is at most worth negative 0.9, but it is actually at most, and I'll just pull this over so you can see it is worth at most -1, just for clarification. Okay, let me mark that one off the list so we don't forget. Okay, next question. Some CNV tracks in DGV do not display inner coordinates. What is the reason for this?
So in the DGV Gold Standard, we should have inner and outer coordinates for all the entries, but in the primary DGV tracks, the way the variant is described will depend on the approach that was used. So for an example, a call that was made using a microarray platform, you'll have only a start and an end coordinate, and they will be defined by the probe that basically was used to call the boundary of the CNV. So they don't extend that out to give you a confidence interval because they don't basically assess that information. So you'll just have a single solid bar without any inner or outer coordinates. And again, there's some of the Sanger-based sequencing approaches will capture the breakpoint perfectly or precisely, and therefore they won't have outer coordinates as well. So it really comes down to the approach or the technology that was used to call each individual variant. I mentioned before, there's a tutorial available on the website as well. And there's actually a page dedicated to highlighting all the different variant types and how they're represented. And that should help with some of the other categories I didn't cover.
Okay. Now the next two questions are coming from Gabe. And Gabe, if I am not understanding your question correctly, please feel free to type in more either in the chat or the Q&A. So the first one, is it better to use the inner or outer coordinates in DGV Gold when following the CNV guidelines for a CNV being common? So I guess the question is really, if you're trying to decide if the CNV that you are evaluating, how well a DGV Gold Standard overlaps it, is it better to use the inner or outer coordinates? If that makes sense, Jeff, if you have any comments on that.
So it may, I guess, depend on the approach that was used by the individual to call their own variants. So if you're using a microarray-based approach, the boundaries may be a little larger, so you could use the outer boundaries. If it's sequencing-based approach, the inner coordinates are probably much more accurate. If we looked at the example I showed, overlapping the Nomad SV entry with the Gold Standard entry, we could see that the inner boundaries of both were almost exactly the same. And the DGV Gold Standard extends a little bit further out. So that's obviously the most highest confidence interval is that inner boundary. So in most cases, it's probably best to use that. But again, depending on the approach that was used to call your individual variant that you're evaluating, you may want to consider both.
Now, this question is from me, and sorry to cut you in line, but so the ranks that you provide, the 1 through 16, 1 being the best, 16 being the worst, is that just in reference to the inner coordinates? It doesn't have any comment on the outer coordinates, is that right?
That is correct, yes. That only references the inner coordinates.
Okay, so you couldn't use that to help you decide how accurate those outer coordinates, because they're really just about the inners, right?
Yep.
Okay. And then his other question, should the 1% threshold in category 4.0 be less for Nomad SV allele frequency, given that it will always be lower than the variant frequency of DGV Gold? And Gabe, I'm not 100% sure I'm following that particular question, so we might just email each other offline. But I think, just in general, when you're looking at frequencies, kind of like in the example that we just went through, there's a lot of things that you should take into consideration before just kind of blindly applying, the rules as written. So you'll want to take a look at how many either samples or alleles were looked at. You'll want to take a look, keep in mind what phenotype are we looking at here, things like that. And if you want to type anything else to clarify that. But we are now at the end of the hour. Oh, you typed something? allele frequency in his example was lower because Nomad is using genotypes. Jeff, do you want to say anything about that?
Yeah, so in this case, in the Nomad example, they had, I think, about 33 individuals that were homozygous for the variant. So that does shift the frequency a bit. But in general, yeah, it's a difference in if every individual in the Nomad SV set was heterozygous for that call, then they're still reporting the allele frequency, which will be about half of what the DGV Gold Standard is as well. So they are different, something to take into consideration, and potentially look at the breakdown of the genotypes at Nomad to kind of reconcile those.
Okay, well, we are now at the end of the hour. I apologize if we did not get to your question, but we can put it in the pool for March 12th. But just a reminder, please log your attendance if you have not already, and we will see you next week when we have Krista Martin with us to talk about considerations with reporting. So thank you.